acids. Nucleic acids was discovered by Friedrich Miescher, a Swiss physician, isolated a new compound from the nucleus of pulse cells, which was quite different from other biomolecules. Therefore, it is named as nuclein. It was found that nuclein had acidic properties and hence it was renamed nucleic acid. Now, how can you define nucleic acid? You can define it that the nucleic acids are polymers of 5 sugar-based compounds called nucleotide. These polymers have high molecular weight and are present in all living things from viruses to mice. Now, there are two types of nucleic acid. One is DNA and second is RNA. Both nucleic acids are linear unbranched polymers. DNA is the polymer of deoxyribonucleic acid and RNA is the polymer of ribonucleic acids. Now we'll discuss them in detail. First we discuss the structure of DNA. So, structure of DNA was explained by James Watson and Francis Crick in 1953 by making model. They proposed that the DNA is a double helical structure. Each helix is made up of 4 types of nucleotide, in which number 1 is adenine, number 2 guanine, number 3 cytosine, and number 4 is thymine. Now, both helices are complementary to each other. That if one helix contains adenine, the opposite or complementary helix will contain thymine, whereas cytosine is complementary to guanine. Each helix consists of 2 parts. How many parts? 2 parts. Number 1 is upright and number 2 is rung. Upright part is made up of deoxyribose sugar and phosphate, whereas rung is made up of nitrogenous base. Both helices are held together by hydrogen bond, due to which zipping and unzipping of both helices occur by making and breaking of their hydrogen bonds. Both helices are opposite in direction, that one chain run from downward whereas the other chain run from upward. एक आ रही है डाउनवर्ड से और दूसरी आ रही है कहाँ से अपवर्ड से, और इनके बीच में डिस्टेंस होता है, द डिस्टेंस बिटवीन टू हेलिक्स रिमेन सेम from one end to another end. Each strand of duplex consists of 10 base pairs. How many pairs? These are 10 base pairs. Now we all know that DNA is a hereditary material. It carries genetic information from parents to offspring in the form of genes. A gene is a part of DNA which has information to synthesis a protein which will work as enzyme. It is a functional unit of hereditary material. The gene gives instruction for developing characters like eye and hair color by producing enzymes. Now let's come to the second type of nucleotide, which is ribonucleic acids. So we have already discussed that it is a polymer of ribonucleotide. The nucleotide contains ribose sugar and one of the nitrogenous base. But in RNA, the nitrogenous base are adenine, guanine, cytosine, and uracil. There is different uracil, but in DNA we have thymine. DNA is a hereditary material while RNA helps in protein synthesis. It helps in protein synthesis. There are 3 types of RNA. Number 1, messenger RNA. Number 2, transfer RNA, and number 3, it is ribosomal RNA. So let's discuss them in detail. So messenger RNA consists of single strand. Its length depends on the size of the gene. It contains information in the form of genetic codes, codon. These codons are basically triplets of nucleotides of messenger RNA which encode one amino acid. It is about 3 to 4% of total RNA in the cell. Now second, which is transfer RNA. The smallest size RNA consists of only 70 to 90 nucleotides. How many nucleotides? 70 to 90. Basically, it is a single-stranded RNA, but it shows duplex at some regions where complementary bases are present. It has anticodons of genetic code as its complementary form. It transfers related amino acid from cytosol to ribosome. They are 60 in number, while human cell contains only 45 different types of transfer RNA. ह्यूमन सेल कितने कैरी करती है? 45 डिफरेंट ट्रांसफ़र आरएनए करती है, जबकि जो ट्रांसफ़र आरएनए जो होते हैं वो 60 इन नंबर्स होते हैं. इट इज़ अबाउट 10 to 20% ऑफ टोटल RNA. नाउ नंबर 3, विच इज़ ribosomal RNA. Ribosomal RNA is present in the ribosome. It has largest size among all three RNA, which is 80% of total RNA in a cell. It is involved in peptide linkage during protein synthesis. अब अगर हम इन तीनों का एक डिफरेंस लें तो मैसेंजर आरएनए जो होती है वो 3 to 4% प्रेजेंट होती है सेल में, जबकि ट्रांसफ़र आरएनए जो होती है वो 10 to 20% होती है. और राइबोसोमल आरएनए कितने परसेंट होती है? It is about 80% of total RNA in the cell, which is which one? Ribosomal RNA. So it is all about today's topic. So I hope you like this video. So do not forget to subscribe and hit the bell icon.